Biochemistry important in microbiology. Carbohydrates, lipids, and proteins, just 3 of the 4 major biochemical groups that are important in all types of organisms. The first major biochemical group to discuss are the carbohydrates. Carbohydrates are typically formed from carbon, to which water molecules are attached. That's why we call them hydrated carbons or carbohydrates. Carbohydrates are often polar or have some part of their structure with either a slight or full positive or negative charge, which makes them tend to be hydrophilic. Polar, some kind of charge, often mix really well with water, which is also a charged or polar molecule itself. Carbohydrates are also called polysaccharides, saccharide meaning sugar. There are various kinds of saccharides. Monosaccharides are the monomers or building blocks of both di- and polysaccharides. Here in this illustration we see 2 very common monosaccharides, glucose and fructose. Notice at the bottom that the molecular formulas, both molecules have 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen. But if you look at the chemical structure, the arrangement of those different atoms of the 3 different elements are distinctly different. So that glucose and fructose, even though they have the same molecular formula, have a different chemical structure, which means they are isomers of one another. Same chemical formula, different chemical structure. The molecular formulas of different molecules don't always tell us exactly how each atom of each element is connected or bonded to one another. And oftentimes, especially with larger numbers of atoms, those different atoms can be arranged in a huge variety of different ways to create different kinds of molecules. So glucose and fructose are just 2 good examples of monosaccharides that are used in the construction of disaccharides and polysaccharides, and they are isomers of one another. Notice in this illustration, not only do we have glucose and fructose, which are isomers of one another, but we also have galactose. All 3 of these monosaccharides are isomeric forms with the same molecular formula, but different chemical structures. We're going to see that each one of these monosaccharides is used in the construction of some very familiar disaccharides. This slide shows several examples of monosaccharides. Monosaccharides can be as little as 3 carbons long and increasing in number of carbons up to maybe 9 or 10. On this illustration we have 4 examples of common monosaccharides. The first 2, deoxyribose and ribose, we will come back to when we talk about the nucleic acids. Notice that with the structure of deoxyribose and ribose, the only difference in the 2 molecules of monosaccharides is located where the hydrogen is highlighted in blue on the deoxyribose and the OH or hydroxyl is highlighted in blue on the ribose. There's really only 1 atom difference between the 2 monosaccharides. In fact, when we take an oxygen away from a molecule, as we have in ribose under item B, and replace it with a hydrogen, we say that that molecule has been reduced. And the prefix that we use in chemistry to describe a molecule that's been reduced, or where we've placed a hydrogen in place of, let's say, a hydroxyl or an oxygen, then the prefix we use is deoxy. So deoxyribose is the reduced form of ribose.
If we take a look at two other examples on the screen, or in the image, glycerol is a 3-carbon monosaccharide, and mannitol is another 6-carbon monosaccharide. So the monosaccharides, which are the building blocks of carbohydrates and polysaccharides, come in a variety of shapes and arrangements.
Is often used as a sweetener in different food products. For polysaccharides, we have starch, glycogen, and cellulose. All three of these polysaccharides are made of large polymers of glucose. In other words, we use glucose, many molecules of glucose. We're going to hook them together with specialized covalent bonds, and depending upon how they're arranged, we can create either starch, or glycogen, or cellulose. Starch is essentially a storage plant for glucose molecules in plants. It's a storage molecule that plants use so that if we need to, and if we have to find places for glucose, we can take that starch and break it down and release the individual glucose molecules to be used as a source of energy. And that's, of course, part of why we eat starchy vegetables like corn and potatoes and rice. Glycogen is the glucose storage molecule in animals. We find glycogen in liver and in skeletal muscle, and again, these are large polymers, but if we need to release individual glucose molecules as a source of energy for metabolism, we simply activate and break down that glycogen into the individual glucose molecules to generate ATP and building blocks that we would need to keep our cells active. Cellulose is also a polymer of glucose made in plants, but instead of having polymers that are sort of branched or large globular polymers, cellulose is a long linear polymer. And in fact, cellulose is the fiber that we find in plants that we cannot digest, but in fact we need in our intestinal tract to help cleanse the intestine. So cellulose is the cell wall material of plants that we use for fiber in our diets, but animals like cows and other ruminants can actually break down and use for food, again releasing those glucose molecules as energy stores.
All of these polysaccharides and even the disaccharides are made from individual building blocks that we call monosaccharides. Glucose is found in most organisms and is the common example of a monosaccharide that we often use with students, teaching them what a sugar is. Fructose is the kind of sweet sugar or monosaccharide found in fruit, so we often do just call it fruit sugar. Galactose is a monosaccharide that can be found in milk. It's actually a component of the disaccharide lactose. Lactose is made up of one glucose and one galactose monomer. Lactose is milk sugar. And then ribose and deoxyribose, again, are monosaccharides, building blocks used to make nucleic acids. When we have two monosaccharides linked together, like glucose and galactose to make milk sugar, lactose, we generally describe that as a disaccharide. If we combine glucose plus one fructose monomer, then we have table sugar. That's the sucrose that some of us like to have in our tea or coffee to sweeten it up. And then maltose is a disaccharide made of two monomers of glucose. And if you've ever had a malted, or if you have ever enjoyed a beverage called beer, malt or malt sugars are often used in those types of beverages or sources of food. And again, it's the disaccharide maltose that gives those foods their distinct flavor or characteristics.
Monosaccharides together, whether we're making disaccharides or polysaccharides, is a glycosidic bond. Now please be sure for exam 2 that you know what a glycosidic bond is. that it is the specialized covalent bond used to link two or more monosaccharides together to create disaccharides and polysaccharides. It's a special name just for this kind of linkage. And notice at the bottom where we have an example of the polysaccharide starch. We simply have long chains of glucose that are linked together with glycosidic bonds, and when we link them together and create a large branching structure, that gives us the polymer polysaccharide starch, all held together by glycosidic bonds.
And just as a side note, it could be that this information about glycosidic bonds might not necessarily be in an exam 2, but is included in other test material for the semester. So please do check with an updated calendar schedule for content and exam material.